Pteridophyta. Introduction. Fern plant grown in the gardens and houses for their aesthetic value is a typical representative of Pteridophyta. Through this, we are all familiar with the group Pteridophyta. These plants also grow wild in shady and moist places, particularly in hills. Once, they covered the whole of the earth several billion years ago. These were found in the form of huge trees then, but slowly they all perished from the earth. Only few representatives are left. Example: ferns, club mosses, and horsetails, etc. Plants of this one group possess vascular tissue, xylem and phloem, for conduction of water, minerals, and form translocation of food. They are mostly herbaceous and grow in moist and shady places. Occurrence. Pteridophytes are usually found all over the world. Some of the plants are aquatic. Example: Azolla, Salvinia, Marsilia. These are among the group of plants which slowly took over terrestrial mode of life. Along with bryophytes, these are called amphibian plants. Other terrestrial pteridophytes usually grow in cool, shady, moist places, both in the hills and in the plains. Some of them grow on trunks of trees and others on soil. General characteristics of Pteridophyta. They generally have a terrestrial mode of life, while a few species are aquatic. They are the flowerless plants like Bryophyta and Thallophyta. Plant body is differentiated into root, stem, and leaves. They do not produce seed, but produce spores, which are borne on sporangia. The sporangia are produced in special structures called sori, and the sori are lodged on the leaves, sporophylls. The spores are homosporous or heterosporous. Vascular tissues are very well developed for conducting water and food. Pteridophytes have alternation of generation. Gametophyte is independent, smaller, and less differentiated. Mature sporophyte is independent of gametophyte. Water is essential for fertilization. Habit. Nephrolepis. The plant represents the sporophyte generation. It is distinguished into root, rhizome, and leaves. Leaves, or the fronds, are the only aerial portions of the plant. The other organs are subterranean. Rhizome. The rhizome is underground. In Nephrolepis cordifolia, special tuber-like structures are seen on the rhizome. Several runners arise from the rhizome, and from these runners, adventitious roots and buds appear, and they grow into new plants. The rhizome is densely covered with brown, soft, and broad scales. Fronds. Leaves. The leaves come above the ground and attain a height of about 15 to 40 centimeters or more. The leaves, when young, are coiled in a circinate manner, like a watch spring. The venation in the pinnae are simple or branched. The pinnae or the leaflets are of two types, sterile and fertile. In sterile leaflets, the veins end in hydatodes or water stomata, but in fertile leaflets, the veins, which are new associated with sporangia, have water stomata. Root. The primary root is short-lived and is replaced by adventitious roots. The roots are thin and black and fix the rhizome to the substratum. They also absorb water and minerals from the soil. Reproduction. The fern sporophyte reproduces by two methods: vegetative reproduction and asexual reproduction. Vegetative reproduction. It takes place by the adventitious buds which develop on the leaves, which form new plants when detached. These buds are known as bulbils. Asexual reproduction. The fern plant is the sporophyte which reproduces by formation of spores. The spores are produced in small asexual reproductive bodies called the sporangia. The sporangia are found in groups on the lower surface of the pinnules. These groups are known as sori, which are situated in two rows on the veins. These sori are pale green when young, but become brown at maturity. Each sorus is covered by a reniform shield called the indusium. The fertile sori-bearing leaves of the fern are called the sporophylls. Leaves which do not bear sori are sterile and called the trophophylls. The sporangia, or the sorus, arise from a cushion-shaped papilla-like protuberance, which is known as placenta or receptacle. Sporangium. Each sporangium is made up of two parts, the stalk and the capsule. The stalk is long, slender, and multicellular. The capsule is a biconvex structure which is filled with spore mother cells. The capsule is enclosed by a single layer of flat, cutinized cells, which forms the jacket layer. Around the edge, the cells are thick inwardly, forming the annulus, which is an incomplete vertical band or ring. On the incomplete side of this annulus, the jacket layer cells are thin and transparent, which form the stomium. The jacket cells at other places are thin-walled. The annulus helps in the dehiscence of the capsule and dispersal of the spores. The diploid 2N spore mother cells of the capsule undergo meiosis and form haploid tetrad spores. Usually, there are 16 spore mother cells in the capsule which give rise to 64 haploid spores. The spores are the beginning of the gametophytic generation of fern. Dehiscence of the sporangia. When the mature sporangium dries, the outer wall shrinks. It strains the stomium and ruptures it. The annulus is curved backward and the spores are exposed. When raindrops fall on the capsule, the annulus suddenly swings forward again, thus throwing the spores forward to some distance. Structure and germination of the spore. The spore are homosporous, minute, somewhat triangular, and brown in color. They are uninucleate and covered by two layers, outer thick, rough, and cutinized exine, and inner thin, intine. The haploid spores germinate on getting moisture. The exine ruptures and the intine protrudes as a short, delicate germ tube. The germ tube elongates and forms a green filament. The basal cell of the filament sends out unicellular rhizoids. The upper cells, after successive divisions, give rise to a small, thin green heart-shaped structure which is called the prothallus or prothelium. The prothallus is haploid and is the gametophyte of the fern. The spores are the beginning of the gametophyte generation. These are minute, brown-colored, uninucleate, and somewhat triangular. Each spore is covered by two layers, an outer thick rough exine and an inner thin intine. The spores are liberated from the sporangia and dispersed by wind. When these spores get moisture, each one germinates into a germ tube, which soon becomes septate and develops into green multicellular filament. The filament, by successive divisions, gives rise to a green, small, haploid, heart-shaped gametophyte known as prothallus or prothelium. Prothallus and sexual reproduction. The prothallus is the gametophyte structure of the fern. It is a small, about 8 millimeters, green, flat, and heart-shaped multicellular structure. At the margin, it has one layer of cells in thickness. There is an apical notch, and the central part is massive and cushion-like. All the cells of the prothallus are rich in chloroplast and can prepare their own food by photosynthesis. From the lower surface arises numerous hair-like unicellular rhizoids which act as roots. The prothallus grow only in moist and shady places. It is concerned with sexual reproduction and bears well-differentiated male and female sex organs, the antheridia and the Archegonia. The sex organs, antheridia, male sex organs, and Archegonia, female sex organs, are born on the lower surface of the gametophyte. Archegonia. The Archegonia are the female sex organs which are found near the notch of the apical cushion of prothallus on its lower side. Thus, it is seen as upside down. Each archegonium is flask-shaped and consists of a base, a venter, and a curved neck. The venter lies embedded partly in the prothallus and contains an egg cell with an egg nucleus and a venter canal cell. The neck is composed of a wall of one cell in thickness surrounding the tubular neck canal. The neck canal contains a row of neck canal cells. On maturity, the neck canal cells disintegrate and produce mucilage and malic acid. Thus, a canal-like passage for the antherozoids is formed. Fertilization. It takes place in the presence of water. When antheridium matures, the antherozoids are liberated. The antherozoids swim freely in water and are attracted towards the apex of the matured archegonium due to secretion of mucilage and malic acid. Many antherozoids may enter the neck canal of the archegonium and pass into the venter. In the venter, only one antherozoid fuses with the egg nucleus and forms the zygote. This process is known as fertilization. The zygote secretes a wall around it and becomes a zoospore. The zygote is diploid and is the beginning of the sporophytic generation of the fern plant. Alternation of generations. In the life cycle of some organisms, there exist two different and specific forms which alternate each other. These two forms differ in form, size, functions, reproduction, and genetic constitution. One of the two forms is more conspicuous. Out of the two forms, one is sporophyte, which is diploid, and reproduces asexually by spores. The other one is the gametophyte, which is haploid and reproduces sexually. The sex organs are well differentiated as male and female and are known as antheridia and archegonia respectively. They produce gametes. Thus, the two forms or generations alternate each other to complete the life cycle of the organism. This phenomenon is known as alternation of generations, which is commonly found in moss and fern. The fern plant exhibits an example of true alternation of generations. The life cycle is divided into two distinct phases or generations. These are the diploid sporophyte and the haploid gametophyte.